sekarang kita akan menginjak pada pembicara yang kedua, ya, yaitu pembicara Profesor Floris Foijer, ya. Beliau pembicara dari Groningen, Belanda, yang akan menyampaikan mengenai chromosomal instability, intratumor heterogeneity, dan juga cancer. Ya, saya akan menayangkan terlebih dahulu bentar, kok hilang. Profesor Floris Foijer, Ph.D. Beliau mendapatkan Ph.D.-nya itu di Netherlands Cancer Institute di Amsterdam, Belanda, ya, yang waktu itu penelitiannya yaitu mengenai protein-protein independent mechanism of cell cycle control. Setelah itu beliau menempuh postdoktoralnya di Harvard Medical School di USA, dan setelah itu juga masih menempuh postdoknya kembali di Wellcome Trust di Sanger Institute, ya, di Cambridge, United Kingdom. Kemudian saat ini beliau menjabat atau sebagai Associate Professor di European Institute for the Biology of Aging, ERIBA, di Groningen, Belanda. Bidang riset beliau mengenai genomic instability pada perkembangan dan juga pada berbagai macam penyakit termasuk kanker, dan beliau juga mempunyai banyak sekali publikasi, ya, lebih dari 50 publikasi. Jadi kalau Bapak Ibu akan mencari mungkin bisa dengan nama belakang beliau, Foijer. Beliau juga menjadi anggota reviewer, ya, di berbagai jurnal dan juga grants, ya, dan juga beliau mempunyai banyak sekali kerjasama penelitian baik nasional maupun internasional. Oke. Saya stop dan saya akan halo Floris, welcome.
Good morning, good afternoon in your case.
Yes, good morning. How is the situation in Groningen right now?
Well, it's getting better. It's we just had a local wave of corona, and we're hoping that the numbers are going to go down, but it's okay. I think it's the same everywhere right now.
Yeah. Okay. Yeah. And now it's summer. I hope you enjoy the nice summer over there. Okay. And your audience here already waiting for your presentation. Yeah, okay. And I think I won't take your time too much. And then I think you can start your presentation. You will share your own screen, or? Okay, yeah.
I'm hoping that you now see my screen.
Yes, yeah.
All right, let me.
Okay, the time is yours.
Thank you very much. Let me see. So it's a great pleasure to be able to tell you something today about chromosomal instability, intratumor heterogeneity, and cancer. So I was very nicely introduced, even though I didn't understand exactly what you were saying, but there were a few things I recognized on the slides for sure. So what I want to do today is to tell you a bit of a background on how the process of chromosomal instability is involved in cancer, and why it's such a dangerous process for cancer evolution. And what I want to start out with is a brief animation that we made to explain the concept of chromosomal instability, aneuploidy, and how that leads to cancer.
We all start as a single cell that divides and divides and divides. An average human lives through 10 quadrillion cell divisions, and in
apologies.
We all start as a single cell that divides and divides and divides. An average human lives through 10 quadrillion cell divisions, and in every division, the cells need to copy the DNA blueprint composed of 46 chromosomes that encodes all the information to make a fully grown human being. When a cell has copied its blueprint completely and ensured that both copies are error-free, the cell starts preparing to divide. Newly copied chromosomes must wait attached until they are all perfectly aligned at the cell division plate. That's when they will be pulled apart by the centrosomes to form two new cells. One unattached or improperly attached centromere will block cell division. Defects in this molecular mechanism will give us two cells with an abnormal chromosome number. These cells are called aneuploid. It turns out that 2 out of 3 cancers have aneuploid cells. We therefore try to understand how these cells behave in comparison to normal cells. But why is aneuploidy so frequently present in cancer cells? Well, let's compare a human cell to a smoothly running machine. When all of a sudden it is presented with a few extra parts, the machine will stutter and eventually halt or run out of control. This is what we think happens to the cell machinery. And thus, with cells that are suddenly presented with an extra chromosome, aneuploid cells will become less fit and in many cases stop dividing or even die. Similar problems occur with an aneuploid cell that has less than 46 chromosomes. Some chromosomes encode genes that are more beneficial for cancer cells than others. Such genes are called oncogenes. Conversely, chromosomes also encode genes that help suppress cancer. These genes are called tumor suppressor genes. However, each chromosome encodes thousands of genes, therefore many oncogenes and tumor suppressor genes. We think that it is the balance between these two that determines how severely the gain or loss of a chromosome affects the fitness of the cell. Whereas gain of a particular chromosome might reduce fitness significantly, gain or loss of another might have a much smaller impact. Either way, aneuploidy has a negative effect on any cell. Let's go back to the machine metaphor. The extra oncogenes provide the machine with extra power, and loss of some tumor suppressor genes takes away some emergency brakes. However, the rest of the machinery is not prepared for these new conditions. Additional adjustments in its blueprint, its DNA, are needed to make the machine actually work. With these alterations in the machine can cope with the extra power and start to divide, and eventually form a tumor. Since we do not fully understand why cells become aneuploid, we try to identify mutations that help aneuploid cells become cancerous. We hope that by identifying these mutations, we will be able to design therapies that reverse their effect. That's what we are trying to achieve in the lab by making large numbers of cells aneuploid in specific tissues, for instance, skin or white blood cells. We found that in mice, aneuploidy is not cancerous. However, one additional mutation of the aneuploid cells is sufficient to go from aneuploid cells to aneuploid cancer cells. When we analyze the resulting aneuploid tumors, we find several alterations to their cell cycle machinery are present. These alterations might help them cope with the disadvantages that aneuploidy imposes on them. We are now carefully mapping these alterations and investigating which of these alterations are crucial for the cells to tolerate an aneuploid state. We expect that our work and the work of our colleagues investigating aneuploidy will lead to new therapies in the years to come. Such therapies would battle aneuploid cancers more effectively and with fewer side effects than most current therapies, and improve the perspective of many future cancer patients.
All right, so that is a brief introduction into the concept of aneuploidy, chromosomal instability, and cancer. And so what I wanted to do today is to explain a bit more how chromosomal instability can arise in cancer, what the consequences of chromosomal instability are for non-cancer cells and cancer cells. And then, time permitting, I will show a few examples from the lab how we try to understand how chromosomal instability leads to cancer and whether we can use that to actually treat cancer patients. So let me start out with a refresher of basic biology, the concept of mitosis. So every single time a cell divides, it needs to segregate its duplicated chromosomes, the chromosomes that were duplicated in S phase, and those need to be distributed over the two daughter cells. And so basically in a mitotic cell, the DNA will condense in the nucleus. And then when it's completely condensed, the condensed chromosomes will start moving to the metaphase plate. And then when they are fully aligned, properly aligned and attached by microtubules that are connected here to the centrosomes, the two poles, the sister chromatids, the duplicated chromosomes are being pulled apart. And in a human cell, that will result in the segregation of 46 chromosome pairs into two daughter cells. So this process of mitosis in cancer cells is very often messed up. And I wanted to show you a brief movie where here you see a cell going through mitosis. You see the chromosomes being condensed, the microtubules in red, and you see that the cells are being, the chromosomes are properly being pulled apart. And in this case, this is a normal mitosis where, again, I'm playing the movie again here. You can see the chromosomes condensed, the microtubules here, and you see the chromosomes being pulled apart. Now, in cancer cells, this process sometimes goes wrong. This is a somewhat lower resolution movie, but what you can see here is that these two nuclei are somehow connected. You can see by the shape of this particular nucleus, there is DNA leftovers that basically connects the two nuclei. And when this happens, there, you know, chromosomes or chromosome pieces can end up in the wrong nucleus. And then you end up with cells with an abnormal DNA content. Now, this is what we call chromosomal instability. And this process of chromosomal instability is a very frequent event in both cancer, but we also think it contributes to aging cells. Now, how does this chromosomal instability arise? So one process that can lead to chromosomal instability is defects in a checkpoint that is called the spindle assembly checkpoint. And in this checkpoint actually measures in a metaphase cell whether all the chromosomes are properly attached to the poles. And so what you can see here in this particular example is that this chromosome pair here is attached by only one pole. And when the cell will segregate the chromosomes, then both copies, the left and the right sister chromatid, will be transported into the left daughter cell, whereas the right daughter cell inherits one copy too little. And so then you end up with an aneuploid cell. Now, the spindle assembly checkpoint is a checkpoint that normally prevents against these type of abnormalities, because what the spindle checkpoint does, it really measures whether every single chromosome pair in mitosis is attached to both opposing poles. And when that is not the case, so in this case, there's microtubules missing from the right pole, then the mitotic checkpoint will be activated and the cell will wait in metaphase until all chromosomes are properly attached, and then the cell will go into anaphase. And so how does this work? Very briefly, the protein names are not that important, but if you zoom into the centrosome, so the kinetochore where the microtubules normally bind to the chromosomes, there's a bunch of proteins, the mitotic checkpoint proteins, and when there is no microtubules attached to the chromosome, the sister chromatid, so one of the two duplicated chromosomes, then these proteins will inhibit a protein called the anaphase promoting complex, and that will prevent the cells from going into anaphase. However, when microtubules attach, then these proteins change their conformation, and then this anaphase promoting complex is being activated that will degrade a number of proteins that otherwise keep the cell in metaphase, and therefore the cell can now exit metaphase, go into anaphase, and the chromosomes can be segregated. So if this process is defective, then the cell will not know whether the microtubules are properly attached, and therefore they might already start segregating chromosomes when sister chromatids are not yet attached by microtubules, and then you might end up with a situation where both sister chromatids go into end up in one daughter cell, and therefore the cells become aneuploid. So another process that can lead to aneuploidy is defects in cohesion, and this is very common in cancer cells. And basically, what cohesion means is there are proteins that are wrapped around the chromosomes that basically keep the sister chromatids together before the cells are going into anaphase. And these kind of wraps that sit around the chromosomes are only degraded right at the end of metaphase. And if these kind of wraps, these rings that sit around the sister chromatids are broken down before the cell goes into anaphase, they can prematurely fall apart. And then two sister chromatids end up being separated from each other, and therefore might end up in the same nucleus. And therefore that leads to aneuploidy. A third process that can lead to chromosome instability is centrosome amplification. And actually the paper that came out yesterday from our lab in genes and development used this particular process to provoke chromosomal instability in the mouse. So when you get additional centrosomes, which are the poles that typically pull the chromosomes apart, when you get more than two, you could imagine that you end up with a cell that has now 3 poles, and then the chromosomes are being directed not in 2 directions, but even into 3, and that will eventually lead to abnormal cells. And that will therefore lead to chromosomal instability. Typically what happens in cells that have centrosome amplification is that the duplicated centrosomes start to cluster. So let's say if you have 3, one of them might end up in one pole, the other 2 on the other pole, and that will lead to an uneven distribution of the microtubules. So the wires that pull the chromosomes apart, and that will lead to chromosome missegregation, as you can see here in this cartoon. And also centrosome amplification is a very common feature of cancer cells. Now, a fourth mechanism that can lead to chromosomal instability is altered microtubule kinetical dynamics. So again, the microtubules are the wires that pull the chromosomes apart. These are polymer molecules, and they're built of tubulin monomers. And if this polymerization and depolymerization dynamics are messed up, which is again also a very frequent process in cancer cells, then the microtubules might not attach properly to the chromosomes, and therefore you will get improper attachment of these microtubules to the chromosomes, and that also will lead to chromosomal instability. Now, another very important mechanism that can lead to aneuploidy is tetraploidization. So now if a cell has duplicated its chromosomes in S phase, then it's meant to go from metaphase to anaphase, as I just explained a few slides before. And in a normal mitosis, you will end up with 2 daughter cells with a normal DNA content. But sometimes the cells fail to segregate, and they kind of get stalled in mitosis. And then the process of mitosis is kind of abrogated. And then instead of segregating the chromosomes, all the chromosomes end up in one big nucleus, a nucleus that has a 4N DNA content, so the double DNA content of a normal cell. And that's what we call a tetraploid cell. And this very often happens, for instance, in cells that have mutations in p53. And when a cell has a 4N DNA content, so a fully duplicated DNA content, the cells will start losing chromosomes much easier, because the cell cycle machinery is not designed to segregate so many chromosomes. So in human cell, 92 instead of the 46. And so that will lead to aneuploidy as well. And so over time, these tetraploid tumor cells will become very aneuploid. So overall, these various mechanisms will lead to chromosomal unstable cells. So summarizing, they can be tetraploidization, when the cell fails to segregate the chromosomes altogether. Centrosome amplification will lead in cells with an unbalanced kind of pooling forces from these microtubules from the poles. The altered microtubule dynamics will result in abnormal attachment of the microtubules to the chromosomes. Cohesion defects will, result in that chromosomes prematurely separate already before metaphase, which will result in missegregation. And the mitotic spindle checkpoint defects will lead to errors in recognizing abnormal mitotic figures, and therefore also to aneuploidy. So, as said, chromosomal instability is a process that will lead to cells with an abnormal DNA content. And so chromosomal instability will lead to cells with extra chromosomes. For instance, in this karyogram that you see here from a mouse cell, the mice have 20 chromosomes, 20 chromosome pair, not 23, so 40 chromosomes in total. But this mouse cell actually has 1, 2, 3, 4 extra chromosomes because there are 3 chromosomes for chromosome 6, 7, 15, and 12. So on the one hand, you will get cells with an aneuploid DNA content. On the other hand, if this process is ongoing, you will end up with cells with an abnormal DNA content, but with different chromosome combinations in different cells. Because, one cell might missegregate chromosome 6, another cell might missegregate chromosome 5, because this is a random process. So you will have cells that have 3 copies of 5, you will have cells that have 3 copies of 6, you will have cells that have 3 copies of 6, 7, 12, and 15. And therefore, if chromosomal instability is ongoing in these cancers, you will end up with cells with different karyotypes. And that will, within a single tumor, and that will lead to cells with different properties, different features. And that is something that we know as, or that is defined as intratumor karyotype heterogeneity. And this is something that could lead, can lead to tumor cell evolution. Now, if we take a step back and we go back to a non-cancer cell, and if we induce chromosomal instability in that, or maybe just aneuploidy, what happens to a cell? And so this is work from the late Angelica Amon, who studied from MIT, and who unfortunately passed away last year. But she did some very important experiments where she provoked aneuploidy in mice, and where she compared the proliferation rate of cells that were normal, so that had just a normal diploid DNA content, euploid DNA content, and cells that had, for instance, 1 extra chromosome. And what she noticed is what on the y-axis you see the cell numbers, and on the x-axis you see the time, that wild-type cells here in open dots always proliferated better than aneuploid cells. And that the aneuploid cells typically were a bit larger. And so the main conclusion from this not so old paper is that aneuploidy confers a growth disadvantage on cells. So actually in a normal cell, aneuploidy is bad news, even though 90% of all cancer cells are aneuploid. If you then investigate what happens to an aneuploid cells by looking at the transcriptome of these cells, what you can see is that aneuploid cells show an upregulation of an inflammatory response and an induction of stress. And what is downregulated are genes that relate to cell division, mitosis, chromosome segregation. And what this tells you is that these cells proliferate less, as I already showed you in the previous slide, and that aneuploid cells are very much stressed. So again, aneuploidy for a non-cancer cell is bad news. So chromosomal instability, what does chromosomal instability do in cancer? And I always like to compare this with the, let's say, the magician who can play card tricks. And so in a normal cell, if you compare the chromosomes in a cell as a deck of cards, in a normal cell, this deck of cards is very evenly distributed. And so a cell that has a syn phenotype, but that doesn't have yet is not yet a cancer cell, you can compare this with really the magician who throws all the cards up in the air and then reshuffles the deck randomly. And what you will then get is probably a deck of cards that will not allow you to win a game. However, the cancer cell somehow has managed to basically get a deck of cards that consists of many aces. And therefore, actually, the cancer cell is cheating by taking the best chromosome combinations and therefore pushing the cancer cells forward by having these favorable chromosome copy number combinations. So the aneuploid cells, over time, we think, select for a favorable karyotype. So chromosomes that help the cancer cells, and select against chromosomes that are not so helpful for them. That also means that this selection process probably takes time. And we really think that the aneuploid cells need to adapt to the aneuploid state to basically convert this growth disadvantage into a growth advantage. And therefore, the cells need an optimal kind of chromosome missegregation rate. If they missegregate too often, the cell can never get this optimal state. If they are too slow, they also can never get to this optimal state. Now, chromosomal instability, as said, will lead to heterogeneity, right? So cells with different copy numbers. And this concept of intratumor heterogeneity is older than the understanding of how karyotype diversity leads to cancer. And here you see an example, an earlier example from a lung cancer where they dissected different regions of the tumor where they actually compared the mutations in these individual regions. And what they found is that these mutations in these individual regions were different. So there was evolution within this tumor. And so we think, or we now know, that actually this evolution also takes place in for chromosome copy numbers. So that some regions will have different copy number states than other, will have selected for different copy number states than other regions. And so this process of, concept of intratumor heterogeneity can actually refer to various types of heterogeneity. And you will hear more, I believe, in later lectures in this course. But very briefly, heterogeneity can refer to the cell types. So a tumor can be made up of, is made up of different cell types. The cancer cells, most importantly, but there are also non-cancer cells. There are immune cells and stromal cells that all support the cancer. It can refer to heterogeneity, regional heterogeneity, basically the different regions that have differences in point mutations, but also in structural abnormalities, pieces of chromosomes that are gained or lost, or whole chromosome abnormalities, so whole chromosomes that are gained or lost. And it can also refer to functional heterogeneity, whether there is angiogenesis, whether there is dying parts in the tumors, or whether there is more proliferative regions than others. But for my lecture today, this whole chromosome and structural chromosome heterogeneity is the relevant type of heterogeneity. One very important concept to remember is that intratumor heterogeneity complicates treatment, because these differences within the tumor mean that different pieces of tumor need different treatments. And therefore, a heterogeneous tumor will also show a heterogeneous response to drug. So one part of the tumor might respond very well, another part might respond less well. And therefore, this is particularly true if you think about heterogeneity for point mutations, structural abnormalities, and numerical abnormalities, because that means that different pieces of the tumor have cancer cells with different features. So one set of the tumor cells might respond to the drug and another won't. So this is very important to take into account, but it's only since very recent that we can measure these regional differences within tumors. And therefore that will in the future allow us to stratify treatment and improve treatment. And so this, that heterogeneity worsens prognosis is very clear from here, where basically you see in blue tumors in patients that have low heterogeneity versus high intratumor heterogeneity. And you can see that the tumors that show high intratumor heterogeneity show a much worse response, both in all patients as well as in patients that are being treated. So briefly, conclusions so far. Various mechanisms can lead to chromosomal instability. These mechanisms coincide in cancer, are often interrelated. Chromosomal instability leads to aneuploidy and intertumor heterogeneity. Aneuploidy changes the properties of cells, and it leads to growth disadvantages. And the chromosomal instability will lead to a whole selection of karyotypes, where some karyotypes are better than others, and those will be selected for in the cancer. But while this process of chromosomal instability is ongoing, it will still lead to intertumor heterogeneity that will complicate treatment and worsen clinical outcome. So how to measure aneuploidy? And so there are various techniques to do this. And there are, techniques that are, much more affordable, but have their disadvantages compared to, methods that are much more expensive, but will give you a much higher resolution result. So the classic way to look at chromosome, to measure aneuploidy is metaphase spreads. And so metaphase spread basically mean that you take cells, you push them, you take them into culture, and you arrest them in mitosis when the chromosomes are condensed, and then you basically put these cells on a slide and you count the chromosomes. So this is simple, cheap, but you need dividing cells, and it's quite labor intensive because every single mitosis you need to count the chromosomes, and you need to, for every single cell, then get to a conclusion whether the cell is aneuploid or not. The main disadvantage here is that it requires dividing cells, and those you typically cannot get from primary tumor samples, because primary tumor samples do not grow so well in a dish. Cancer cell lines do, but primary tumor samples don't. So an alternative then is interface FISH, where you take fluorescent probes that you then hybridize to individual chromosomes. And so you basically take a probe that recognizes, for instance, chromosome 4 here in red, and another one that recognizes chromosome 10 in green. And then you can see that this particular cell has 3 copies of each. So therefore, this cell is aneuploid for both of these chromosomes, whereas this cell has 2 copies of each and is a normal cell. So this is, a method to also measure aneuploidy in non-dividing cells, and it's quite cheap to do. But it actually, these dots, sometimes, a piece of dust will also give these type of dots, and therefore it gives a bit of background. And you can only measure 2 to 3 chromosomes per a cell, even though there are 23 pairs in a normal cell. So therefore you can only assess aneuploidy for a few chromosomes. A third method is comparative genomic hybridization. This is where you take a piece of normal tissue that has a normal DNA content. You take a piece of tumor tissue that has the abnormal DNA content. You label the DNA of the control in one color. You label the tumor DNA in another color, and then you chop it into pieces and you hybridize it to a chip that has the whole genome on it, and then you compare the colors. And so on these chips, you will have little dots that represent probes, for instance, for a gene that is not amplified, and there you will get equal hybridization of the test DNA or the tumor DNA and the control DNA. You'll get a yellow dot. If you have a red dot there, that means there is more, for instance, tumor DNA. That means that in the tumor that was gained. When you see a green spot, that means there was more in the control, and that means that that DNA was lost in the tumor. So that is a very powerful way to, at high resolution, look at the whole genome. You can do it for all types of cells. It is relatively expensive, and you will only get the average karyotype, so you cannot measure heterogeneity. Another method is flow cytometry. By flow cytometry, you can measure the DNA content by just taking, by adding a DNA dye to the cells, and then you can basically, by a histogram, you can plot the cells based on their DNA content, and you will get cells that have a G1 DNA content or a normal DNA content, and a few cells that have the double DNA content, which probably are the cells in the G2 phase. And so when cells are aneuploid, you will get a wider distribution of the cells in D1, because some cells will have an extra chromosome and therefore they have a bit more DNA. So this will be a bit wider. But this is a super insensitive method. And it can measure maybe, cells that are tetraploid, but it is very weak to recognize cells, within, that have 1 or 2 extra chromosomes. There, it's not sensitive enough. So then a last method, and you will also hear more in a later lecture, I think in the next session, about single-cell DNA sequencing and a beautiful application of that, is single-cell DNA sequencing. So what you can do is you can actually take a piece of tumor tissue, and you can chop it into single cells that you can then seed into a multi-well plates, where every well will get a single cell. And then in a fully automated fashion, and this is a method that was developed partly at our institute, you can make single cell whole genome sequencing libraries. And so then for every single well, we can perform a sequencing reaction, and then we can assess the DNA content of the individual cell. And so what you will then get is a genome-wide map of the single cell, where you can compare the DNA content per chromosome. And so what you see here is a zoom in of some of this data, where you can see, for instance, basically the signal for chromosome 1. And what you can see is that if you look at the numbers here, that the signal for chromosome 2 is about 50% higher than for chromosome 1. And that for chromosome 4, the signal is actually 2 times as high as for chromosome 1. And using a tool that we developed in the lab that is called Aneuploidy, you can then basically annotate the whole genome, saying, okay, chromosome 1 has 2 copies, chromosome 2 has 3 copies, chromosome 4 has 4 copies. And so this method has become a very powerful tool in our lab, and we've been doing this with many, many collaborators to map for non-dividing cells from primary tumors the complete DNA content. And therefore, if you do this for multiple cells in a tumor, you can map the DNA content of cells. And so here you see an example of that, where you see the DNA content of a healthy control sample, where now every line here, and there's about 50 lines here, represents the karyotype of a single cell, where green means there's 2 copies and purple means there's 1 copy. So this means that all the chromosomes have 2 copies except for X, where there is 1 copy. And this means this was the healthy genome of a male participant. And what you see here—
5 minutes left, okay?
Yes.
Sorry, yeah.
Yeah, thank you. So what you see here are 3 examples of tumors where you can see that there is many aneuploidies. And what you can see here is that, for instance, for chromosome 4, 9, 14, and 15, all of the tumor cells that we sequenced have either 3 or 4 in yellow copies. So this means that all of these 3 tumors have selected for an extra copy of chromosome 4, 9, 14, and 15. So this is what, an example of how the tumor cells have selected for a particular karyotype. However, what you can also see is that for many other chromosomes, it's much more random. So you see that some of the cells, select for an extra copy of 1, but by far not all of them. And if you look for chromosome 2, this tumor, half of the tumor cells are selected for an extra copy of 2, but not so much for tumor 2 and tumor 3. And so this is where you can see that even though all of these tumors select for chromosome 4, 14, and 15, and 9, not all of them do that for the others. And you can see that within the tumor, there is a lot of heterogeneity too. So let's now say, if you were thinking from a tumor's perspective, and these are untreated tumors, let's now imagine that the tumors would, that tumor cells that are sensitive to the drug treatment that you would treat these cancers with, have 2 copies of chromosome 8, but they might become resistant if they have 3 copies of chromosome 8. You could imagine that this tumor might already have 3 cells here that are predisposed to become tumor resistant, to treatment resistant, whereas these 2 tumors don't have that yet. But if this process of SIN, of chromosome missegregation, is ongoing in these tumors, also these tumors might have an opportunity to actually become drug resistant. And so therefore, this intratumor heterogeneity that is fueled by the process of chromosome missegregation can lead to drug resistance. And this is why measuring chromosome instability and the resulting intratumor heterogeneity is so very important in cancers. And this is really something that only has become available in the last few years. And we really think that this method will therefore become very, very important in the future to stratify tumor treatment. And since I'm running out of time, and since I will end my presentation in one more slide, because of that, I invite you to download our paper that came out 2 days ago in Genes Development, where you can actually see how we have used single-cell sequencing to map chromosomal instability, and where you can see these recurrent chromosome copy number changes of 4, 14, and 15 in cancer, and that these actually arise in multiple cancers that are even arising when we provoke different ways of chromosomal instability in mice, driven either by centrosome amplification or by mitotic spindle checkpoint defects. But in summary, the methods that I've just described to you all have their advantages and disadvantages. So there are methods that basically require a cell to go into metaphase, and therefore you would need the cell to be able to divide. And so these are typically the cheaper ways to look at aneuploidy. And that is a very powerful tool when you want to assess aneuploidy in cancer cell lines that grow well in the dish, that grow well in the lab. However, you know, when you want to measure chromosome instability in primary tumor samples, you either are limited to more expensive methods like CGA, where you can measure the average DNA content, but not yet, you know, heterogeneity. So therefore, aneuploidy at the single cell level, or you are limited to methods that can only measure 1 or 2 copies at the same time. And with that, I would like to end my presentation and give some time for questions.
Okay, thank you, Professor Foijer. Yeah, thank you for the very clear presentation, how you explain the step to cause the CIN, and then the CIN cause the heterogeneity. And I saw already several questions from the audience. First, from Dr. Zulham from the Faculty of Medicine, North Sumatra University. Yeah, the question is, it looks like that the aneuploidy-related genetic instability is closely related to the microtubule or tubulin expression and end growth. Is it possible for a single cell presenting different microtubule kinetochore growth rate among chromosomes and develop aneuploidy-related cancer and its end?
Yeah, so there are various mutations known in cancer that will lead to an altered microtubule growth rate and shrinking rate, and so it's therefore and there's actually drugs cancer drugs that interfere with that. So there are drugs, cancer drugs that interfere with microtubule growth rate, and that will lead to aneuploid cells in cancer cells. And because aneuploidy is detrimental, by increasing the chromosome missegregation rate, even in cancer cells, you can kill cancer cells. So yes, these altered expression of these type of proteins will lead to more severe aneuploidy. I'm rereading the question to make sure that I'm answering the question properly.
Sorry, Floris, maybe you can close your
presentation
slide, and then we can showing the question to you. Okay.
Yeah.
Okay. Yeah. Thank you, Yaya. Can you make it full screen? Yeah, so the question that I I delivered is the third question, and maybe now we can go to the first question. It's about,
So so in case of cancer, what causes a cancer cell? So you're referring here to, let's say, defects in the mitotic spindle checkpoints. And so, one way for a cancer cell to get defects in the mitotic spindle checkpoint is by overexpressing. Many cancer cells overexpress components that are important for this mitotic checkpoint protein. So I showed you this kinetochore image with some of the genes, like the BUP genes and the MAD genes. And when these are overexpressed, this mitotic spindle checkpoint gets deregulated. And then the cells can no longer signal properly when chromosomes are not properly attached. And so in our mouse models, we did the opposite. We inactivated genes in the mitotic spindle checkpoint. And then also what you will get are cells that fail to measure where the chromosomes are properly attached. And when they fail to measure this, they will go through mitosis with abnormalities, and then they will become aneuploid.
Okay. And the second question, we still have like 3 minutes.
So how do cells select for advantages? So this is a very good question that we partly understand, and so I'm going to refer again to this issue of this genes and developments papers that actually came out 2 days ago. Next to our paper, there is a back-to-back paper from the lab of the late Angelica Amon, and they did a very beautiful experiment to prove how a single chromosome gain can allow for selection of a chromosome. So as you probably are very much aware, cancer cells select for, growth advantages by deregulating, genes. And this deregulation can be the result of, epigenetic deregulation, so upregulation of a gene. It can be a mutation to a gene, but it can also be a copy number change. Now, in this other paper that is back-to-back with ours, what they have done is they have shown why lymphomas that are very similar to the lymphomas that we find in our paper always select for chromosome 15 amplification. So I showed you that also our tumors select for chromosome 15. One of the genes that we have known for a long time to be on chromosome 15 is c-MYC. And c-MYC is one of the most well-known transcription factors. And what they have shown is if you inactivate the c-MYC gene on chromosome 15 in mouse tumors, that then the tumor cells no longer select for chromosome 15 amplification. What they also did is they took the MYC gene and they placed it on chromosome 6. And chromosome 6 in these lymphomas is rarely gained. And now when they moved the MYC gene from chromosome 15 to chromosome 6, they could show that now all of a sudden chromosome 6 was gained in these tumors. Now, and this answers your question, because basically the selection mechanism is the genes that are on the chromosome. So 1 or 2 genes on a single chromosome are powerful enough to drive a chromosome copy number change. If the MYC gene goes from chromosome 15 to chromosome 6, the tumor, instead of selecting for chromosome 15 will select for chromosome 6. So we really think that the selection is because there are genes, oncogenes that are beneficial for the tumor, and the tumor will select for that. Because the extra copy will give you extra expression of that gene, and then the cell can, benefit from that.
Okay, yeah. For the question number 4, I think it's a bit related with the question that you already answered in number 2 and number 1, but maybe you can add some for it. Actually, yeah.
So does aneuploidy cause cancer? Yeah, I think it relates a bit to my previous answer, right? So, and so this is actually what we try to understand in the lab. So we know that cancers select for particular karyotypes. So the karyotype of 4 next. So in this particular lymphoma, extra copy of 4, 14, and 15. We've also shown in the past that if you do this in a different tissue, for instance, liver, you get a different karyotype. So then all of a sudden chromosome 13 becomes important, for instance. So it matters which chromosome is missegregated, and it matters even for the tissue. So one tissue will select for a different copy number change than another one. And then, you know, chromosome 13 gain is going to be not so beneficial maybe in a lymphoma cell. So if a lymphoma cell gains chromosome 13, it will be selected against, and it will respond with a growth delay. So we think you might want to think about, you know, a karyotype as something that gives the cells a particular fitness. So 99.99% of all the karyotypes will reduce the cell fitness, and only 0.01 or less of the karyotypes will give the cells a fitness advantage. And therefore, you know, by random missegregating cells, most cells will lose fitness and disappear, and only few cells will gain fitness. And what we try to understand in the lab is, you know, what defines this fitness, and are there features in cancer cells that all aneuploid cells share and that we can exploit to kill them. But for that, you know, unfortunately, I didn't have time today, but make sure that in the next year to keep an eye out of you know, the type of things that we will publish, because we have some exciting papers to share there. Maybe in a future course, I can explain a bit more about
yes.
it.
Okay, thank you, Floris. Actually, there are 2 questions more, but because the time is up, we can send the question to you, and then later you can answer, give the written answer, and then we will deliver to the audience. Okay.
Right.
Thank you very much for the presentation and also the discussion. So
thank you very much for having me, yeah. And yeah, have a great day.
Okay, have a great day. Bye.
Bye-bye.
Thank you. Yeah.
Thank you so much.
Okay.